All right. Welcome everybody to our KOL learning series for Bayesian stats, and very happy and excited to welcome Steve Ruberg to present to us today. So just briefly, going to share a little bit about Dr. Stephen Ruberg, who received a bachelor's degree in mathematics at Thomas More College, Miami of Ohio, and PhD statistics from University of Cincinnati. Just a little bit about Steve from a personal perspective. I have worked with Steve in the past. I've always found him to be very knowledgeable, very helpful, and always very pleasant to listen to, and just educational in so many different levels. He is published extensively in a wide variety of statistical topics related to preclinical and clinical drug development. And his current research interests include estimands, subgroup identification, Bayesian methods for clinical drug development. So we're so excited to have you with us today, Steve. So with that, I believe that you will share your slides.
Yes.
And take it from here.
Yes, thank you very much, Melissa, for the invitation, and thank you very much for the invitation to speak to this wonderful group. All right, all right. Can somebody just confirm that everything is showing up just fine?
Yes, we can see and hear you.
Okay, great. All right, well, off we go. So this is a pretty bold statement, I think, the epistemological superiority of Bayesian inference over frequentist inference. But I think it's in our quest to discern what is likely to be true, or infer what is likely to be true. So I'm going to give you my perspective, a bit about my journey into Bayesian thinking. I never took a Bayesian class formally in my entire life. I've never done a Bayesian analysis. I can't run proc MC or bugs or any JAGs or any of these things. But I have come to learn and understand and appreciate Bayesian thinking and how it can be useful, even if I don't know the mathematical or computational mechanics behind it, the power and the utility and superiority of thinking from a Bayesian perspective. I also realized that, what can I say to this group, the DIA Bayesian Statistics Working Group, who has taken Bayesian classes and do Bayesian analyses and are deeply engaged in, doing many Bayesian things in your companies, in this working group, who've studied this deeply. So I must say, I'm a little pensive about what I have to say. Some of this might come across as like, oh, gee whiz, we already know that. I hope that some of the stuff I present is maybe using some different stories and analogies. It will help you communicate to non-statisticians or even other statisticians about the Bayesian perspective, if you will. So if some of this sounds very familiar, well, my apologies. I hope the stories and the analogies are new enough that it can help you. But I think there's a few things in here that might be new, that represent some of my thinking as it's evolved from a frequentist background to a Bayesian background. So I'm going to tell some stories, give examples, and again, help you perhaps with how to communicate and teach others about Bayesian methods. And maybe there's a few new ideas in here for you as well. Okay, so some history. Again, maybe some of you know this, but this has come to me a little bit more recently that Abraham de Moivre, in the History of Statistics, 1711, he published his book, The Doctrine of Chances, which is subtitled A Method of Calculating the Probabilities of Events in Play. And basically his notion was, the model, the truth, flipping a coin, rolling a die, or whatever it is. And probability is merely the frequency of events, the number of times that an event occurs that you're interested in. And it's particularly when n is infinite, we can say that the probability of heads on a fair coin is 1/2, even if we never actually observe that, in a sequence of flips or in an experiment or whatnot. And I must say that whenever I saw the title of Thomas Bayes' essay, an essay towards solving a problem in the doctrine of chances, I did not understand or fully appreciate that he was actually referring to de Moivre and his book, The Doctrine of Chances, right? In which Bayes says, well, we don't know the model. All we know is the data, and we have to infer what the model is, the underlying truth or whatever, right? So he was I always thought that when I read this title that it was just a generic phrase of the time, the doctrine of chances, a different word for probability or phrase for probability. But he was actually referring to a problem in Demoivre's book where he said, well, I don't know how to solve the inverse problem. Like if I have the data, how do I know what the model is? He said, I don't know how to solve that one. And that's what Bayes wrote this essay towards solving a problem in the doctrine of chances, quite literally what he was doing, right? Whatever he wrote was so important. We know that Richard Price actually found this, published it, read it to the Royal Society. And I look at, it was read on December 23rd, 1763. So Price and the Royal Society, they must have felt like this was substantial enough or important enough that it had to be read 2 days before Christmas. Now I'm reading into this a little bit. I don't know the exact history, but I find it kind of striking that this finding, this essay, this solving this problem was sufficiently important enough to call a meeting of the Royal Society 2 days before Christmas to have the essay read. And in the introduction to the essay, Richard Price says, hey, I want everybody to know that Thomas Bayes has solved the inverse probability problem. And he says, I know of no person who has shown how to deduce the solution of the converse problem to this, namely the problem, or how Demoivre framed the problem of probability. Namely the number of times an unknown event has happened and failed being given to find the chances that the probability of its happening should lie between two named degrees of probability. In other words, what's the underlying parameter? How close can we get to the underlying parameter given we've observed this, these events happening or not happening, etc. So anyway, just a little bit of history. Maybe some of you guys are steeped in all of this and you knew that Thomas Bayes' essay was literally referring to Abraham Demoivre's book from several decades earlier. But this is some stuff that's kind of new to me and I find it fascinating, and I'm going to continue to think about how the whole story unfolds in the history of statistics and mathematics. For my story, the first story on my journey came from this kind of thought experiment that was given to me by Brenda Gatos, who many of you know, was at Eli Lilly with me. But she said, suppose there's a PowerPoint jackpot of $300 million, and here's what I'm going to do. If I win, there's a 90% probability that I am going to share with you, all right, the audience, all of us that are here, right? But there's a 10% probability that I'm going to keep it to myself. So if I win, I'm going to put 10 pieces of paper in a hat, mix them all up at random, pull one out. If I draw the numbers, 0 through 9, or I'm sorry, well, 1 through 9, then you share in the winnings with me. But if I pull out the number 10, I'm going to keep it all to myself, right? And so when I teach this to people, I use this example when I talk about Bayesian statistics, and I say, how likely are you to receive a share of the winnings? Even those who have no training in statistics realize that the answer is either C or D. It's something small that they know that they're not going to get, there's a very low chance that they're actually going to get any winnings. And that's because they intuitively know something about conditional probability and the key word if I win, right? Because there's a very low probability of winning, we know that there's very little chance that you'll get a share of the winnings. And the formal solution that I lay out for people is to say, well, the probability that you actually get money from me is the probability I choose to share if I win, but I have to take into account the probability that I win, the probability of this condition, right? So even though it's a 90% probability of sharing if I win, the probability of winning is so small that your intuition was correct. The probability of you getting a share of the money is exceedingly small. And sometimes I like to say very, 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 very small, okay? Just to be clear. This is an unconditional probability, as we know. This is a conditional probability. We have to understand when we condition on something, what's the probability of that something occurring? And of course, this plays into conditional or traditional frequentist power versus the probability of study success, right? And so I may go on to teach about probability of study success or Bayesian power as maybe a more relevant quantity to understand when you're designing a trial rather than power, because I can make the probability of me sharing my winnings with you 99%, 99.9%, and there's still a very small probability that you will get any money. And it's the same way with power. I can make the power as large as I want by making the difference that I condition on as large as I want. So anyway, probability is hard. Conditional probability is even harder. I learned about Bayesian power, probability of study success, and I thought, what other concepts should I be thinking about and learning about with Bayes or whatever? Along my journey, I created this thought experiment for myself in thinking through this, and it's become an invaluable teaching tool that I've used for non-statisticians and for statisticians alike, right? I found that there's many statisticians who've taken lots of Bayesian classes per se. They know the mathematics inside and out. But even those folks, when I presented this thought experiment, have said to me that it's a very powerful thought experiment. In fact, Brad Carlin, who many of you know, when he first saw this example, this thought experiment, he said to me it was the most insightful example he's ever seen that's highlighted the distinction between Bayesian and frequentist thinking. So I took that as very high praise, and I like sharing this and using this, right? So the thought experiment goes like this. Suppose I have 10,000 coins, 9,999 of which are fair, and one is biased. The problem is this: I'm going to draw one coin out of the bag at random, mix it all up, draw it at random. I'm going to flip that coin repeatedly and tell you the result, okay? And you're going to tell me when you decide whether I have the biased coin in my hand or not. Now, of course, as soon as I flip the coin and get a tails, you know I have a fair coin in my hand. So the only interesting part of this thought experiment is how many consecutive heads would you need to see before you'd be willing to say, bet that I've got the biased coin in my hand, right? And so I do this process. I say, if I flip the coin and I tell you it's a head, would you bet that I have the biased coin, yes or no? And of course, everybody says no. Two heads in a row? No. 3 heads? No, I'm not willing to bet yet, Steve. But as I march up the line to 5, 6, 7, 8 heads in a row, I ask people to raise their hand if they think I have the biased coin in my hand. And hands start going up when we start to get to 6, 7, 8 heads in a row, 9, 10, many heads are going up, and I work my way through. And usually by the time I get to 20, everybody's hand is up. Although there have been times where a few stalwarts have held out and said, no, I need to see more than 20 consecutive heads to bet that you have the biased coin in your hand, right? So I say this is a problem of inference. The kind of decision rule that's in your mind is how many heads, consecutive heads, do I need to see before I'd be willing to bet, right? So we can couch this as a frequentist hypothesis test. The coin is fair, or the coin is biased. The probability of heads is 0.5 or 1 under the null or alternative hypothesis. The probability of n consecutive heads, given that I have a fair coin, given the null hypothesis is true, is simply 0.5 to the nth power. N is your choice based on your risk. It defines the significance level of the test. I'm speaking to many statisticians. You all know this, right? But I explain this to my non-statistical audience, and I say, suppose your decision rule was observing n equals 10 consecutive heads. That's when I'd be willing to bet that Steve has actually got the biased coin in his hand, right? And so you would be thinking of this, the probability of n consecutive heads, 10 heads under the null hypothesis, the probability is very, very small, 0.00009. And so you'd say, wow, the significance level of the data, the p-value is very small. Therefore, I'll reject the null hypothesis, and I'll say it's highly likely that Steve has selected the biased coin with a p-value less than 0.001. Okay, seems quite logical to most people when I present it, right? Then I say, well, but null hypothesis significance testing is kind of like proof by contradiction, right? You want the alternative hypothesis to be true, or you want to understand whether the drug works, whether that's true or not the null hypothesis that the drug doesn't work, right? So you're usually looking to contradict or prove the null hypothesis false, or to say, I believe the alternative is true. So what we really want to evaluate is the probability that the alternative is true given the observed data, or that the null is false given the observed data. And the question is, how many consecutive heads are needed to bet that I selected the biased coin? In other words, what's the probability that I pulled the biased coin from the bag? Or what's the probability that I got the biased coin in my hand given that I've observed n consecutive heads? And I want that to be greater than 50%. That is, the odds are in my favor if I were to make a bet, right? So the probability of me winning would be greater than 50%. How do I find out what n is? Well, thankfully, we have Bayes' theorem, and you can say, what's the probability of the biased coin given I've observed n consecutive heads, or the probability the alternative hypothesis is true given n? And from Bayes' theorem, as formulated by Laplace and mathematically in 1812, it's this formula, right? And if we plug in the numbers, the probability of n consecutive heads under the alternative is 1. The probability of the alternative is 1 in 10,000. Probability of n under the alternative or under the null is 0.5 to the n. The probability the null is true is 9,999 over 10,000. And if we can calculate that probability for any value of n, but if we did it for 10, we get a probability of 0.093. And I emphasize, this is not a p-value. This is actually a direct probability statement about the probability that I'm holding the biased coin in my hand, given that I've told you there's been 10 consecutive heads when I flipped that coin, okay? A very low probability, less than 10%, right? So it's such a highly unlikely that I've selected the biased coin. I mean, there's a 90% chance that I do not have the biased coin in my hand. You would say, I am not going to make that bet. Okay? So now my audience is usually pretty puzzled, and I say, how do we get into this mess? The frequentist approach says we should bet on Steve having the biased coin, overwhelmingly positive, but the Bayesian approach says, no, overwhelmingly negative. Don't make the bet. You're very likely to lose. Okay? Well, you know, we have these two different perspectives. The frequentist approach, probability of seeing n consecutive heads under the assumption that the null hypothesis is true, versus the converse, right, which is the Bayesian approach, taking into account that we have a bag of 9,999 fair coins and only one biased coin, right? When you put up the p-values, if you take the frequentist approach and put all the p-values, and again, here's 10 consecutive heads, the 3 zeros, 0.00097, etc. Here's all the p-values. And when you look at 13 and 14 consecutive heads, what do you see? Let's take a look at that in more detail. Well, with 13 consecutive heads, the p-value is 1.2 in 10,000, right? If we take it out to the 10,000th place, 1.2 in 10,000. With 14 consecutive heads, the p-value is 0.6 in 10,000. And so the probability that I pull the biased coin from the bag is 1 in 10,000. And, whoops, I hit the wrong buttons. So it's actually more likely to get 13 consecutive heads with a fair coin than it is for me to pull that one biased coin out of a bag of 10,000 coins. But 14 consecutive heads is a little less likely for me to, or it's less likely to observe 14 consecutive heads than it is for me to pull that one coin out of the bag. In other words, if I observed 13 consecutive heads, I'd be more likely to say, I think it's still possible the null hypothesis is true. Whereas with 14 consecutive heads, you'd say, well, I think that's a more rare event. That's a very unusual event, even more unusual than Steve pulling that one coin out of the bag. And therefore, I'll go with the notion that he's actually got the biased coin in his hand. That's a more likely outcome given the data that I've observed, 14 consecutive heads, right? And I explained p-value is a conditional probability. It's conditional on H not being true. So why do we accept it at face value? Recall the lottery example, right? For any conditional probability, we need to ask ourselves, what's the probability that the condition is true, right? It's just logical. That's how you need to think about or evaluate any conditional probability. And so what's the probability that the null hypothesis is true in this example? Well, it's nearly 1. It's 9.9999, right? Probability that the null hypothesis is true. And so if the probability the null hypothesis is true is very, very large or close to 1, then you need a whole lot of evidence to reject it, okay? You need overwhelmingly convincing evidence, not just 5 consecutive heads, not just 10 consecutive heads, but maybe as many as 14 consecutive heads, right? Which I'm dare say that I've never seen that. Maybe you've never seen that if you've done coin flipping experiments to see 14 consecutive heads. But that's the strength of data you need to kind of overcome this condition that, well, what's the probability the null hypothesis is true, right? More on this later, okay? More on this later. So the two perspectives, you know, we have the probability the coin is biased given the observed data, right? Is the Bayesian perspective that we've been talking about, and I say that's what we really want to know. And when you look at the Bayesian probabilities, drawing the coin out of the bag before I ever flip it, the probability I have the biased coin is 1 in 10,000. But with one head, if I apply the Bayesian formula for calculating the posterior probability that I have the Bayesian coin, it's now increased ever so slightly. With one scrap of evidence, one heads, I now have a slightly higher probability or belief that I have the bias, or you should have that I have the biased coin in my hand. And of course, you can map this out with each flip of the coin. When you have 10 heads, there's the 0.09 probability that I have the biased coin. And you can fill in the table all the way through. But what you'll notice here is with 13 heads, there's the probability that I have the biased coin is 0.45. With 14 heads, it's 0.62. So with 13 consecutive heads, the probability I have the biased coin is a little less than 50-50. That is, the odds are not in my favor, but with 14 consecutive heads, the odds are in my favor, 62% probability of winning. And in fact, with the p-value, you can say, well, this is where the switch is flipped from things being in my favor, less likely or more likely. But the Bayesian probability gives you an actual quantity to say, well, 14 consecutive heads, not only are the odds in my favor, but I can tell you precisely what those odds are, or probability of me winning is, it's 62%, okay? So the Bayesian result is much more informative and much more accurate for answering the question of interest, right? So this is what I was saying is that, you know, the even odds for the bet are 50%. With 13 heads, it's less likely you want to bet. With 14, you have a higher probability of winning. The odds are in your favor, right? So when I'm done going through that and explaining to people, I say, okay, let's do that same thought experiment with 100 coins, of which 99 are fair. Same experiment. And then I ask, how many heads would you have to see before you'd be willing to bet? And while many of the students can't give me a precise answer, the general answer is, well, I think it's less than 14. I need less evidence. There's fewer coins in the bag, so somehow intuitively it seems to me I need less evidence to convince myself or to be willing to bet that you might have the biased coin in your hand. And indeed, again, we can go through the same exercise where the prior is 1 in 100, right? And fill in this table with these Bayesian posterior probabilities. And you can see here now with 10 heads, when you have only 100 coins in the bag, there now is a 90% chance, 91% chance that I have the biased coin in my hand if I reported to you that there's 10 heads, okay? And if you look, what I like to say is that note that the p-value never changes. When you put the whole story together, the frequentist p-value, the context where there's 10,000 coins, the context where there's 100 coins, when you put it all together, what you realize is that when the p-value flips from greater than 1 in 10,000 to less than 1 in 10,000, that's when the probabilities flip in your favor. The same is true for this situation. When the probability goes from greater than 1 in 100 to less than 1 in 100, the p-value, that's when the probability goes from not being in your favor, 39% chance probability of winning, to 56% probability of winning. The odds are in your favor, okay? But what I say, so you make a different decision, right? But what I like to say is that the p-value never changes regardless of your prior knowledge, because you make an assumption about, I know the truth. The null hypothesis is true, okay? And so it never changes. And I challenge people to think that, is that a good way to make inference? That you would always make the same inference regardless of the context? And of course, people realize the answer is no. So I say, yes, it's a very important lesson to learn. For the same data, that is the same evidence, you arrive at different conclusions, different decisions based on your prior knowledge, okay? So that's an important message that we need to share in this whole thing. And this example helps to highlight that, okay? So I like to say you cannot interpret a p-value in isolation. It's a conditional probability, and you have to have some knowledge or notion of how likely the condition is to be true, okay? I'm not saying ban p-values. I'm not saying get rid of them or they're useless. I'm just saying they're only part of the story, and you can't interpret them in isolation because it's a conditional probability, right? It's the probability of our test statistic exceeding a critical value under the assumption that the null hypothesis is true. And you should always, I tell people, don't just look at the p-value, have some notion of how likely is it that the null hypothesis is true, or what was your prior belief before going into this experiment or this trial or whatever about how likely the null hypothesis is true, okay? So as you all know, but I like to teach, the frequentist is the probability of the data given the null hypothesis. The Bayesian is the probability of the hypothesis given the data. And what I like to tell the students is, if you think this is just a subtle little mathematical statistical twist or flip or whatever, it's like a stunt, or it's just something for you statisticians to think about or worry about or whatever, I like to use the analogy. I say it's as different as the probability of cloudy given rain versus the probability of rain given cloudy, okay? And I tell the audience, think of any examples, practical examples like this, rain and cloudy. Substitute your own words in there and then reverse them and tell me if those two probabilities are the same or have the same meaning or the same interpretation or whatever. And if your answer is no, as it should be with this example of cloudy given rain versus rain given cloudy, you realize these are fundamentally different notions of inference about what is likely to be true, okay? So as I like to say, traditionally statisticians have been selling the probability of the data given the hypothesis, p-values. We all know this. P less than 0.05 is kind of the gold standard, and even if it's not the gold standard, p-values are still quoted routinely and completely and thoroughly throughout many, if not almost all scientific papers. But what we really want is the probability of the hypothesis given the data, a Bayesian probability, to quantify the likelihood of a hypothesis. Scientists, when I talk to them, when I talk to clinicians, what they really say is, I have some idea about whether my drug works or this molecule works or not. I'm going to do an experiment. And now that I've seen the data from that experiment, I want to reassess my belief, my knowledge, my understanding about whether the drug works or not. And as we all know, that's very much in the Bayesian paradigm of prior, data, posterior. And if you talk to them, that's what they really want. But we say, statisticians wrongly have been selling for decades, well, probability of data given the hypothesis, that's close enough. And as I tell my students when I teach them, no, cloudy given rain versus rain given cloudy are two very different things. And so I like to say this is the first great bait and switch that statisticians have pulled on scientists. Scientists want the probability of the hypothesis given the data. We've done a bait and switch, and we've reversed that conditional probability and say, yeah, but it's good enough, it's close enough, it still is meaningful. And proof by contradiction can still be meaningful, but it's not the answer that scientists want. In fact, that all of us want. Okay, we've taken an easy calculation that was easy to do in the day of Sir Ronald Fisher nearly 100 years ago and said, this is close enough, and we've actually switched on scientists for this, okay? All right, so it's the first great bait and switch. The second one, which is the topic for another conversation, is using intent to treat to estimate a treatment effect. But that's a whole different conversation and lecture on estimands. But that's another bait and switch. All right, I'll stop there for 1 minute, take a little drink, and see if somebody else, see if anybody has a comment or a question. Melissa, are you managing the chat room or the online?
I am monitoring the chat, and there are no questions in the chat right now.
Okay. This is all pretty rudimentary, I know, and I hope that the way I'm conveying it or the examples and stories I'm using can be helpful to you when you try to communicate with other statisticians. I will say the example that I just showed you with the point in the bag, I was talking one time to a statistician who knew Bayesian statistics inside out, could do all the calculations, all the multiple integrations, whatever the case may be, etc. But he was still very much a frequentist statistician of give me the p-value to make my decision. And when I did the coin in the bag example, and I got to the last slide and I said, you know, and the p-value never changes. You would always make the same decision based on the p-value, whereas the Bayesian, you would make different decisions. I remember him sitting back and thinking for a moment and saying, man, I've been doing Bayesian statistics for a long time mathematically, but I've never thought of it quite this way. And he said, I have to think about this. This is kind of jarring to me because I you know, it's a different perspective or whatever. So anyway, I just share that with you as well. All right, Melissa, anything in chat or anybody raising their hand?
There are some comments talking about there are good illustrations and everything is very clear. So I think that people are enjoying it and engaged so far. Good.
Okay. Well, we're going to keep digging deeper here. Here's another story on my journey, and that's the next thought experiment is suppose that we're doing a diagnostic test. 5% of the population have an ALK gene. We develop a diagnostic test with 95% sensitivity, meaning that out of 20 patients who have the gene, our diagnostic test on average would detect 19 of them and would have one false negative, and it has 95% specificity, meaning that for every 20 people who do not have the ALK gene, the diagnostic test will correctly identify them as negative, but there will be one false positive finding on average, 1 out of 20. Okay, no diagnostic test is perfect. Sensitivity and specificity describe, how likely you are to have true positives and true negative findings. There will always be some false negative and false positive findings. But the key is when an individual patient comes into the clinic and you don't know whether they're ALK positive or negative, and you do take a blood sample and do the diagnostic test to test for the gene, if the result comes back positive, the question is what's the probability the patient is actually ALK positive, right? So I pose this to my students and I let them guess, 95%, 25%, 100%. I get numbers that range all over the place, right? So I go through this example and I say, when you're developing and designing an assay, right, your conditional probability, now that you know what conditional probability is, moves in this direction. You start with patients that you know are positive, run your diagnostic test, and see how many patients come out positive on the diagnostic test. In the same way with negative, you start with a group of negative patients and you do the diagnostic test and see how many come out negative. And that's how you define your sensitivity and specificity. It's the probability the diagnostic test is positive if the patient has the characteristic, or diagnostic test is negative if the patient does not have the characteristic. That's specificity. But of course, when you actually are interpreting an observed result and you don't know what the patient characteristic is, the unknown truth, right? The conditional probability moves in the other direction. All you observe is whether the diagnostic test comes back positive and negative. And of course, we have this inverse probability problem. What's the probability the patient has the characteristic if the diagnostic test is positive? And that's what we call positive predictive value and negative predictive value. And of course, that's what the patient and the physician want, right? Hey, I don't know whether I got the disease or the characteristic or whatever it is you're trying to measure, All I know is the test is positive. Now, doc, tell me, do I have the disease or not? Do I have the characteristic or not? Or the test is negative. We saw a lot of it, an explosion of this with the COVID disease when it emerged, the pandemic emerged. You know, people were arguing and debating over false negatives and false positives and all these kinds of things. What they really wanted to know is what's the positive and negative predictive value of a diagnostic test, okay? So if you work out this example, and I do this arithmetically just to make it easy for students, I say, suppose I have 2,000 patients randomly selected from the population. I know that 5% of them have the ALK gene, which means 95% don't, which means in my sample of 2,000 patients, 100 will have the ALK gene, 1,900. Again, using simple arithmetic, recognizing there would be random sample variation, but using simple arithmetic to make the calculation simple. That means 95, the sensitivity is 95%, 95 out of 100 of these patients would come back positive, 5 would be a false negative. For the 1,900 who are negative, 95% would test negative from the diagnostic test. That's 1,805, and there would be 95 false positives. 5% of 1,900 is 95. And the positive predictive value is the number of true positives over the total number of positives you get back from the diagnostic test. Remember, in reality, all you know is the results of the diagnostic test. There's 190 positive diagnostic tests out of these 2,000 patients. 95 of them are true positives, so the positive predictive value is only 50%, even with a diagnostic test that has very high sensitivity and specificity. The negative predictive value, converse of this, easy to calculate, 99.7%. So if you take this diagnostic test and the ALK gene is, the diagnostic test is negative, you can be nearly certain that you do not have the ALK gene. But if it comes back positive, it's a coin flip as to whether you have the ALK gene or not, right? So even with a low prevalence, the idea is with low prevalence, the positive predictive value is low, okay? But if you were to do a retest, right? What if we did a retest, all those 190 patients that were positive from the first test, what if we retest them, okay? Well, you now have an enriched population. And with any diagnostic testing, right, you go from simple and inexpensive to more and more invasive and expensive, right, each step, but you're identifying a higher prevalence population that may have the disease or condition or whatever it is that you're dealing with. So if we took the 190 people who were positive in that first ALK diagnostic test, and we reran them through and simply repeated the test, there's now a prevalence of 50% of the people in that population that are positive, okay? Now, again, just to make the numbers easy, I'll say, suppose we have 2,000 patients. If you do the arithmetic and fill it in, you now see the positive predictive value is 95%, and the negative predictive value also remains high at 95%. So the moral of the story I tell the students is, if you just test positive once, don't necessarily believe you have the ALK gene. But if you test positive twice, there's a 95% chance that you truly have the ALK gene. If you test positive once in the first test and negative in the second one, there's a 95% chance that you do not have the ALK gene. That is, the first test was a false positive finding. And of course, if you test negative on the first test, then you're not even going to bother to test again because there's an extremely high probability that you truly are negative, okay? This is how drug development works as we go from phase 1 to phase 2 to phase 3. We're constantly identifying an enriched population of drugs that may work, all right? But our priors are usually should be pretty low early in drug development, and they increase as we accumulate more evidence through phase 1, 2, and 3 drug development. All right, the key messages for diagnostic testing: sensitivity and specificity are the focus of assay design and development. Sensitivity is equivalent to power. Specificity is 1 minus specificity is equivalent to the size of your hypothesis test. Positive and negative predictive values are the essential outputs for interpreting results. Everybody knows this. Every physician who does diagnostic testing knows this. PPV is what matters to physicians, to patients, to the healthcare system, to everybody, right? Now, PPV and NPV are dependent on the underlying prevalence of the characteristic of interest. Prevalence is the prior. Literally, it's if you take a random sample of people in the population, what percentage of them, what proportion of them have the disease or the characteristic of interest. So going into doing the diagnostic test, that's your prior, that's your probability that any patient selected at random has the characteristic, right? And what I learned through this whole process is that the formula for positive predictive value is literally identical to Bayes' formula that I've shown back on slides 19 and 25, right? Most people don't realize this, that I've run into. This is a little bit of a surprise to them. It was a surprise to me when I first saw this, that it really is exactly the same formula, all right? So the question is then, a clinical trial is really just a very sophisticated diagnostic test, right? The unknown truth is, does the drug work or not? And I'm doing this very sophisticated assay called a clinical trial, and I'm going to do a hypothesis test as like my diagnostic machinery to reject or not reject the null hypothesis, right? Sensitivity and specificity are the power and the significance level. PPV and NPV are the Bayesian posterior probability that the treatment meets whatever critical success factor you have. And the PPV and NPV are dependent on the prior probability that the treatment meets your critical success factor, right? Whatever that might be, right? So in phase 2, if you have the probability that a drug works across the industry, the average is about 20% when you're entering phase 2. About 20% of the drugs entering phase 2 make it to the market. And if you had a moderately rigorous phase 2 clinical trial design, maybe a false positive rate significance level of 10% and a power of 80%, let's just work through this same example. If I observe a clinical trial result that's positive or negative, just to make the arithmetic easy, if this were the scenario for 2,000 different drugs across drug development in the industry over a number of years, you would end up with numbers in the table that look something like this. Simple multiplication. The positive predictive value of our clinical trial diagnostic test, if it came back positive with a statistical test that had these characteristics, there'd be a 2/3 probability that the drug truly works, that it is a true positive finding, right? A 95% chance if the trial result, if you don't reject the null hypothesis or meet critical success factors, 95%. This is a simple way to do it. Of course, there are much more sophisticated Bayesian approaches for doing this kind of modeling, et cetera. But I like to show this example because clinical trials are a direct analogy to a diagnostic test. And people are surprised that you could have a positive study, phase 2 study, but still have I would say a pretty good chance that the drug works, but there's still a 1 in 3 chance that your clinical trial might fail. 20% is the prior. These positive and negative predictive values are the posterior probabilities. So the conclusion on inference is this. We all understand PPV is the proper metric for evaluating the likelihood of an unknown condition to be present or true in a diagnostic test. And a clinical trial is a direct analogy to a diagnostic test. I mean direct, not approximate, direct. So then why do we not routinely use a Bayesian posterior probability to interpret a clinical trial? PPV, which is a Bayesian probability, is uniformly used to understand whether a patient has a characteristic or not when they have a diagnostic test done. So why don't we routinely use a Bayesian posterior probability to interpret the outcome of a clinical trial? Significance level and power are important to the design of the trial. Bayesian posterior probability is essential and the only thing that logically makes sense when interpreting the result. Again, p-value is only part of the story. Well, the answer to the question is we should, obviously, we should. Okay, I'll take a pause again because I know I'm preaching to the choir, but I wanted to share you some of my thought and development, my maturity along the way, moving from frequentist to Bayesian thinking, and how I try to lead other statisticians and non-statisticians on this journey. I'll stop. Melissa.
Yeah, Steve, we do have a couple of questions, I think, to a previous example that you had walked through. The first question is, if good statisticians still make their decisions according to frequentist p-values, could it be that the conditional probability in your example is assumed 1, whether that is true or not?
Yeah, I mean, I'm going to talk a little bit about this later, but yeah, there's the notion of if you assume the null hypothesis is true, in one sense, you're saying the probability the null hypothesis is true is 1, okay? Now, here's the logical fallacy of that. If the probability that the null hypothesis is true is 1, then why are we even doing an experiment? Why are we even doing a trial? Why are we even calculating a p-value or trying to make a decision that would reject that hypothesis, right? If your assumption up front is the null hypothesis is true, then that's the end of the story if the probability of that is 1. So in some sense, I even see there being kind of a logical inconsistency of let's assume absolutely completely the null hypothesis is true. And then when we get a p-value that's sufficiently small, say, oh, we now reject the null hypothesis. Well, obviously you then didn't, if you're willing to reject it, then you obviously didn't really believe 100% that the null hypothesis is true, okay? So that's kind of how I view that part of the story. But I'm going to talk a little bit more about that later. I hope that answers the question, or I hope that makes sense to some people.
Okay, we'll hear more about that in a little bit. Another question was a request. Would like to hear your thoughts on how to explain the coin example to non-statisticians if you don't know how many biased coins are in the bag, since that reflects real world scenarios.
Yes, that's a question that comes up all the time in that example. I shouldn't say all the time, comes up once in a while. But Steve, we don't know how many coins are in the bag. And I go, yeah, you're right. But if I brought out a bag of 10,000 coins and I brought out a bag of 100 coins, I bet you could give me some idea, some guess, or we could do some prior elicitation from experts who know something about the size of coins and the volumes of coins. And we could say, yeah, the bag with 100 coins is definitely smaller than the bag with 10,000 coins. And we could probably come up with some approximations to that scenario. So like I said to my students, my non-statisticians, you're right. We don't know the prior for sure, like we do in the coin in the bag example, but that doesn't mean we know nothing. It doesn't mean, that we have no opinions or no belief, or we have no data about what our prior knowledge about a drug working or not might be, okay? So you're right, we don't know the prior, but that's how I answer the question of, yeah, but I can tell the difference between a bag of 10,000 coins and a bag of 100 coins.
Okay. All right, those were the questions that were in the chat so far.
Okay, good. I hope those are coming close to answering your questions, and hopefully maybe a little later we can dig in on those a little more if need be. But I hope those were satisfactory answers. Maybe if those were satisfactory answers, you can chat in a thumbs up or something. If not, you can chat in a thumbs down, and we'll dig in a little deeper later. All right. Okay. So how do we get out of this mess? How do we get out of this mess? All right. Well, I like this framework that I read about from Richard Royal about 25 years ago, but he says there's really 3 inferential questions. What does the data say? And a p-value is a partial or poor answer to that. We know p-values can be influenced and made to be as small as we like with a given large sample size. So it's not necessarily a good answer, okay, but it's an answer. The next question is, what do I believe, which requires incorporation of prior information. And then what do I decide, which requires a utility function. Now, going back to the coin in the bag example, another interesting question I get from some students once in a while, they go, hey, Steve, what's the bet for? If it's for $1, I may be willing to bet you when I see 10 or 12 or even 14 coins, knowing what I know now. But if the bet's for $1 million, I might have to see 20 consecutive heads in order to be 99% sure that, you have the biased coin in your hand, because I can't afford to lose $1 million and I don't want to risk it, right? And I always tell them, That's a great question. What's the bet for? Because in any decision about rejecting the null hypothesis or believing the drug works or doesn't work or whatever, we need to incorporate the value of that decision. Okay, so so those are the three questions, and I love that framework. And when I've done some consulting, when I was at Lilly and talked to people, I used to walk through this and help clarify kind of each step of the process as to decision making. What do you do? I think the problem is what the frequentist world and what the scientific world has done is the p-value serves all three purposes. We reject the null hypothesis, so we believe the drug works, and we're going to decide to approve the drug. Okay, that way madness lies. I think to use the p-value to do that carry the whole load. So, question one: what do the data state? I like to say a p-value is only part of the story, as I've shown. Right? We have prior knowledge about whether the null hypothesis is false. We have a new experiment where we generate a p-value. And of course, I like to say everybody wants this. Think of the probability I get money from the lottery example. If Steve wins the lottery, of course, the frequentist focuses on the central piece of this question, right? Not the whole story. And so what we need to do is say, what do I believe? And I'm going to show you something I've learned. I presented this at the JSM meeting to the folks who are at the Bayesian working group meeting there. And I was pleasantly surprised that in one sense, people hadn't seen this. So I felt good that I was presenting something new, but I was a little disappointed that more people didn't know about this. But here's how I think about this in a very quick, easy way of assessing outcomes and posterior probabilities. So let P0 be the probability, prior probability that the null hypothesis is false. So that's the answer to question one. Okay, oh, I'm sorry, let P0 be the prior probability. So that's our prior. Let P be the p-value from the test of that null hypothesis. Okay, that's the answer to question one, right? I'm sure many have heard of Bayes factors bound, which is this function of the p-value given here. But what I found most people aren't familiar with is a publication by Tom Selke and others that gives the upper bound on the posterior probability that the null hypothesis is false given the observed data. And here's the formula for that, which holds under some pretty general conditions. And what this has in it is the prior odds is expressed here. The data is captured by the Bayes factor bound, or the p-value, which you can think of the p-value as the ultimate test statistic distillation of the data into a single number. And this gives you the bound, this formula gives you the bound on the posterior probability that the null hypothesis is false, okay? That you can reject the null hypothesis. Now, I've used this quite extensively, right? But I'll give you one quick example. Suppose there's 100 potential predictive biomarkers that could be important for a new treatment, all right? I was literally involved in this when I was at Lilly looking at rheumatoid arthritis. People were looking at a broad panel of biomarkers, and then we wanted to do a hypothesis test on each one to see if people who had this biomarker produced a differential treatment effect. The observed p-value from one of the hypothesis tests was 0.0001, okay? And the scientists and I looked and said, well, hey, even for the most conservative adjustment of that p-value, the Bonferroni approach, which the scientists knew about and knew that it was conservative, we still get an adjusted p-value of 0.01, which is highly significant. And there was this belief, hey, eureka, we've discovered a novel biomarker-defined subgroup where our drug works exceptionally well, right? Well, I'd ask the question, are you sure? Suppose further that our belief, I say, what was your prior belief that there was a biomarker that would be predictive for our drug going into this trial? Well, after hemming and hawing around, 20% came out that, yeah, we would have thought maybe going in, there's a 20% chance that we could find a biomarker out of this set of 100. I said, okay, so the prior that at least one null hypothesis is false is 20%, 0.2. The prior that all null hypotheses are true, that none of these biomarkers are predictive, is 80%. Yeah, okay, we agree with that. We believe that. I said, okay, I'm going to put a uniform prior per biomarker. I'm just going to simplify this and say, take the 0.2, divide it by 100, and say my prior is 0.002 for any given biomarker. If you have no a priori knowledge about which one of these biomarkers, I'm just going to distribute my prior uniformly. I know these biomarkers are correlated, et cetera, but I'm going to take this kind of Bonferroni conservative approach, if you will. So if we have a prior p-value or prior of 0.002 on any biomarker and a p-value of 0.001 for one biomarker that was significant, highly significant in this panel, if we use the formula for calculating the upper bound based on the prior belief and the p-value, this is Bayes factor bound here, we get the Bayesian posterior probability that the null hypothesis is false is less than or equal to 44%, 44, right? Less than a coin toss. So we went from a very conservative assessment and adjustment of the p-value, still leading to a very significant result, indicating, yes, we found a biomarker, to a less than a coin toss chance that this biomarker is actually predictive of who will respond to our treatment, okay? All right. Now, again, I know there's more sophisticated approaches for subgroup identification. I point to some work by a former colleague of mine, Lei Shen, Jim Berger, and his graduate student at the time, to assessing all of this. But this simple formula has really played out quite well in my experience. That it's been very valuable and it's been enlightening. It's been useful in describing the credibility of results in moving away from p-values, even multiplicity adjusted p-values, to Bayesian posterior probabilities. Okay, so go read the paper and others related to it. I found this to be extremely useful. I've got some blogs out there on my blog, analyticsthinking.blog, where I've applied this to a drug called dalcetrapib, where they said they identified genetic subgroups. I did and their assessment was based on p-values and adjusted p-values and all that stuff. I said, I don't believe you. The Bayesian posterior probabilities are quite small that you've actually found a genetic subgroup. There's another drug, fluvoxamine, for COVID-19, an antidepressant that was tested and had some positive results in a trial for reducing the effects of COVID-19 disease. How come I have COVID-9 there? It's COVID-19, right? And when I did my analysis and said, well, my prior would be quite small, then an antidepressant would actually be helpful for COVID-19. So I wrote this all up in a blog, et cetera, and said, the Bayesian posterior probability is quite small. I don't think really fluvoxamine works in COVID-19. Well, late breaking news. In July, right, the definitive dalcetrapib study that was done, so the people who found this purported genetic subgroup said, we're going to do another $400 million outcome trial in this specific genetic subgroup. And lo and behold, they just published their results a month ago. Dalcetrapib did not significantly reduce the risk of recurrence of primary endpoint ischemic cardiovascular disease or events, right? It doesn't work, right? FDA, there were some other studies done with fluvoxamine, some phase 3 studies done in Brazil, and FDA came back and said, no, we don't believe that there's enough evidence for emergency use, for even emergency use authorization for fluvoxamine. Data are insufficient to conclude this. The FDA did not take a Bayesian approach formally, but if you read this memorandum, they're basically saying all the other evidence, our priors and blah, 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 really don't kind of add up to substantial evidence. Again, if you read my blogs, you'll get more of those stories in more detail, okay? So a p-value is literally only part of the story. It's this central part of the story. And we really want the whole story. And I believe that this relatively simple formula actually tells a pretty good overall story. And Tom Selke, Jim Berger, and some others have investigated lots of studies and reports of p-values, and they have reported that the p-value bound, the bound here is reasonably sharp, okay? There are some assumptions and some things you might, but at least they say this is a reasonable approximation. And in my experience, I think that's a reasonable belief or conclusion as well. So here it is, your reasonable or your run-of-the-mill statistician says Always use Bayesian thinking when assessing clinical trial results so you can quantify how believable the results are. When I read medical papers, when I read the results of clinical trial papers, I first ask myself, what would be my prior that this kind of drug would work in this disease state or in this patient population or in this genetic subgroup or biomarker defined subgroup, right? What is the chances that the next drug that's being studied for the A beta hypothesis in Alzheimer's disease, what's the chances of it working, or what's my prior? And I say, given all the failures in Alzheimer's disease, my prior would be 5% or less, maybe even 1%, given all the failures, right? So the next study that reports positive results in a p-value of 0.02, I take that in the context of my prior and might say, I'm not completely convinced if my prior was low enough. I'd at least do the probability calculation using this formula, since I don't have access to the raw data in all the formal mechanisms and machinery for calculating a Bayesian posterior probability. A simple formula seems to work reasonably well. Okay? Further investigations into p-values. Maybe I should stop there for a second and say, before I do further investigations into p-values, some other thoughts or questions?
Yes, I'm glad you paused. There was a couple of comments here and a couple of questions. A useful example to consider when a case of rare AE is noted in a treatment arm and the product ends up with a warning in the label. Presenting Bayesian arguments could help inform this. It was a general statement that was made.
I think that's true. But we all know the playing field isn't level for efficacy and safety. You know, for efficacy, you have to have substantial evidence. That's defined in law. For safety, I think we all feel that for safety, it's irrational to say, I don't need as much evidence to put a warning out there or to put something in labeling, right? It could be helpful in deciding whether you get a black box warning or a statement or whatever to say, what's our a priori knowledge on this? But again, safety and efficacy, not a level playing field, more biased toward reporting potential safety concerns. And I think there's logical societal healthcare reasons for that. But you're right, it can be helpful.
Here's a question. I'm wondering whether any of your non-stats trained students were clinicians who asked you about your trial's framework and how to reconcile priors with the ethically necessary for randomization concept of clinical equipoise among all trial arms. Were there any? Yeah. And then how did you answer? And thank you for sharing your thoughts.
Yeah, that's a good question. Are we imbalanced when we say, oh, there's equipoise, there's equal belief that this drug works or doesn't work? My answer to that is, and that Bayesian thinking is at odds with that, to say, well, my prior is that the drug works, et cetera. My answer to that is, I hope every phase 3 trial that's done, there is not equipoise in whether the treatment works or is no different than placebo. I hope that your companies are not investing tens, if not hundreds of millions of dollars and many years of clinical trial research and exposing many, many patients, thousands, hundreds, if not thousands of patients to a drug for which you could honestly say, I have no idea whether this drug works or not. It's a coin toss as to whether it's better than placebo or not, okay? So I don't think, I understand the notion of equipoise and randomization in clinical trial ethics. But I also know that we don't invest hundreds of millions of dollars in doing a phase 3 trial if we don't have something inside us that says, we believe this drug works, and we think we're at the point where it's now ethical to expose hundreds or thousands of patients for 6 months, 1 year, 3 years, whatever, to confirm the degree to which it works, et cetera, okay? So that's how I answer that question. I don't think equipoise exists when you're actually doing some of these confirmatory, when you're doing almost any confirmatory phase 3 trial. That answer may be satisfactory or unsatisfactory. There may be ethicists and philosophers who would give other different arguments, but that's kind of my thinking.
One other question or comment in question. I think your answers are reasonably good, but in the regulatory setting, the sponsor still needs to demonstrate efficacy in a sufficiently powered study. Phase 2 is not sufficiently powered. The reason that agencies ask for two independent trials, positive trials, perhaps in part to try to prove the conditional probability is close to 1.
Yeah, I mean, two adequate, well-controlled trials. Replication, I think, is a very, very important thing in science in general. Yes, phase 2 trials are not adequately powered in many situations, or not definitive. That's why we do phase 3. But it doesn't mean we can't use that data and other data external to our phase 3 clinical trials. Perhaps there's phase 3 trials of other drugs in the same disease state, in the same mechanistic action class, right? There's no reason we can't use that information to help us interpret our results from our phase 3, the phase 3 trial that you were planning on doing, okay? I've heard a good analogy. So suppose people are doing a 1 mile race, and after the first 2 laps, you know, there's somebody in first, second, third place, whatever the case may be, right? Well, what if the rules of the race were, okay, that's great, but now who's ever in the lead, in second place, whatever, everybody's got to stop, and we're going to wait for everybody to catch up. And now you have to start over and say, okay, now run the last 2 laps and let's see who wins, right? I think that's a reasonable analogy. It's an interesting analogy or story to tell, to say, you know, if you're running a 1 mile race, somebody who has the lead at the half-mile mark should be given credit for having the half-mile lead, and they shouldn't have to wait for everybody to catch up to run the last 2 laps to prove that they're the fastest or the best runner, right? Can't we use what they've already done and build that in and say, overall, here's the best runner for the 1 mile race? So in any case, those are some thoughts that I have about using other information and some of the arguments.
Okay. Here's another comment. Many thanks. So a reconciliation may be presumed prior is informative, yet evidence to date at the stage of designing a randomized trial is not sufficient to contradict clinical equipoise.
I think I understand that statement, and I think I agree that stating a prior is not at odds ethically with the notion of equipoise. I think that's what the comment was.
I think that's what it's saying as well.
Yeah. Okay, let me move on. We'll keep plowing ahead here, okay?
Yeah.
Future investigations into p-values. So I have another blog on this, is what does p less than 0.05 mean anyway, right? So using the formula that I just showed you, if you have a prior of 0.3 and a p-value of 0.05, your posterior upper bound is about 50%, right? About a coin toss, okay? So if you have a phase 2 trial where your prior is 0.3 maybe, and you get a p-value of 0.05, there's only a 50-50 chance that you've actually identified a true positive. That is a treatment that really works, right? And I've heard it said by Bob O'Neill when he was at FDA. I've heard other regulatory people say this. I've heard other statisticians say it. A p-value of 0.05 is not very strong evidence against the null hypothesis, okay? And indeed, you might look at this and go, yeah, I guess so, if you take this perspective. But then I say, well, wait a minute, if my prior is 0.7 and I get a p-value of 0.05, my posterior upper bound is 0.885 that the null hypothesis is false. Gee, that's pretty strong evidence. Again, whether the p-value is 0.05 or not, you can't interpret that in isolation. You can't say it's not strong evidence because it depends on your prior, right? It might be enough evidence against the null hypothesis. And if you fill in this table using the formula that I've been sharing with you, you'll notice that here's the increase in posterior probability, that you went from a prior of 0.1 to a prior of 0.21 with a p-value of 0.05. So you've increased your belief that the null hypothesis is false by a relatively small amount, okay? And it changes depending on your prior. It gets larger here when you're up to 0.4, and then it gets smaller, et cetera, right? So a p-value of 0.05 does not move the evidentiary needle very much, is how I like to say it, right? At most, you get about a 0.2 increase in probability for your posterior probability, right? I've written a whole blog on this and looked at different scenarios, but it does give you some idea why people say a p-value of 0.05 is not very strong evidence against the null hypothesis. I think what they really mean is it doesn't move the evidentiary needle very far. If you start very high, you'll end up a little bit higher, but not a whole lot higher, right? If you start very low, you'll end up a little bit higher, again, but not very much higher. So it doesn't move the posterior, it doesn't move from prior to posterior very much. Which leads me to this notion of the probability of a false positive finding that I said we'd get to later, right? Recall this, right? We got to look at the conditional probability. So the probability to reject H naught given H naught is true. If we design an experiment, we have a significance level, alpha, a false positive rate. After the data is collected, we calculate the significance level. The convention is to use the smallest p-value for which we would have rejected the null hypothesis if we had observed, given this observed test statistic, right? Well, Fisher talked about the p-value as evidence to be considered whether something's significant or not. The Neyman-Pearson lemma, they used, this as a decision-making criteria. You balance type 1 and type 2 errors using sample size, to make decisions about whether to reject the null hypothesis. But our modern convention, is the p-value as both is something at least that maybe came before Layman in 1986, but, he says it's good practice to determine not only whether the hypothesis is accepted or rejected at a given significance level, but also to determine the smallest significance level, the significance or probability or p-value at which the hypothesis would be rejected for the given observation, okay? Now, here's my problem with this, is that we all know when we design an experiment, a hypothesis test, we define the test statistic, the size of the test, the rejection region, that all has to be done a priori, right? You figure that all out, you write that, you put that in stone. Once you collect the data, you can't change that, right? You got to stick with what you said. This is ICHE 9 pre-specification. It's just good statistical practice, good scientific practice, no matter what, right? So what happens when we run the analysis, we run some statistical model and compute a test statistic, and we compute the p-value. And the p-value is nothing more than the ultimate test statistic. The p-value is a one-to-one and onto mapping via an inverse probability function, okay? So the p-value really conveys no more information than the test statistic value itself. It's just that it's easier to say the p-value is 0.01 than it is to say my chi-square statistic on 6 degrees of freedom is 13.7, okay? Those are mathematically equivalent statements. The p-value is kind of easier to understand. But where I have a problem is now that I've observed the data and the test statistic, right, to compute that p-value and say, oh, gee, if I would have done a hypothesis test at 0.01, I would have rejected the null hypothesis. And to somehow convey that now the probability of a false positive finding is even smaller than the 0.05 I originally said, well, now you're looking at the data and going backwards and saying, here's the size of the test that I could have done to reject this hypothesis. I'm just a humble statistician. Eric Lehman's a giant in our field, but I struggle with that philosophy of saying, now that I've observed the test statistic, I'm going to go back and say, here's the hypothesis test I wish I would have done and the size of that test, et cetera, okay? So there's this other philosopher of science and statistician, his name's Gigerenzer, and he called this the silent hybrid solution, where we conflate the significance level of the test, alpha, with the significance level of the data, the p-value. And people think that the p-value is now the significance level of the test and the probability of making a type 1 error, right? So the practical question is if you design this experiment with a 0.05 p-value, conduct the experiment and observe 0.01, that is, you reject the null hypothesis, a positive finding. The question is, well, what's the probability that this is a false positive finding, right? Or the probability of a false positive finding is what's the probability that the null hypothesis really is true, given that I've observed a p-value of 0.01, or the converse, 1 minus the probability the null hypothesis is false, given a p-value of 0.01, right? That's the real question of interest, and that's decidedly a Bayesian formulation of the problem. what's the probability of the hypothesis given the data? Now, typically when we design a test, we might have a significance level alpha of 0.05 and a critical value of 0.1 or not 1.96, and we call that the significance level of the test, right? But the significance level, we might and should be thinking about, well, what's the probability the null hypothesis is true, okay? And if we say had a prior distribution of effect size that looked something like this, you might say, well, there's 30% of the area of this distribution where the null hypothesis is false, and 70% where we would say the null hypothesis is true. The treatment doesn't work or it's actually harmful, right? So if we actually did this, the probability to reject H naught given H naught is true times the probability the null hypothesis is true, you might say, well, we're actually, the size of our test under this condition is actually 0.0175, okay? All right, that's really our significance level of the test. And the question might be, well, if I really want the significance level of the test to be 0.025, what value of X do I need here to get to 0.025? Well, the answer to that is if X is 0.035 times 0.7, the size of the test is 0.025, and the critical value is 1.80. That is, if we have some belief the null hypothesis is false, we need less evidence in order to make the conclusion or reject the null hypothesis in the frequentist kind of paradigm, okay? So once again, I'm developing these thoughts and trying to think through this, but it fits into that notion of if you have some belief the null hypothesis is false, then you should need less evidence in order to reject that null hypothesis. And you could do these simple calculations if you wanted to get some idea about, well, what is the traditional size of the test and the critical value that you should be using in order to convince yourself that, yes, I have substantial evidence or enough evidence to reject the null hypothesis. Now, of course, I understand this is quite controversial in that people don't think this way, but I hope as I've been telling this story that this is not too far-fetched, or this is not out of the question, crazy, ridiculous, that it does make some sense, okay? So this is why I think it's difficult to reconcile frequentist approaches and Bayesian approaches. When I hear people talk about frequentist properties of Bayesian approaches, I bristle a little bit at that phrase because, as we've said, the frequentists say the null hypothesis is true, one, absolutely certain, can't change my mind. Whereas the Bayesian approach says something, the probability is less than one, okay? All right, so I'm gonna stop there for a second. This is my latest idea thinking about positives of false positive findings and kind of the inappropriate use of a post hoc p-value and how people wrongly interpret that as the size of the test they could have done, and therefore the probability of a false positive finding is somehow related to the p-value. So I'll stop there we got a little bit of time left. I'll stop and take a couple comments or questions and then go to the final epistemology slide.
All right, thank you, Steve. There was one other question that kind of slid in just as we were wrapping up the previous sets of questions. I'm going to go back to that. Should we distinguish between two scenarios? One, where you are looking for independent evidence, for example, a phase 3 confirmatory trial versus, or two, a second scenario where you are looking for accumulative evidence, such as patient or clinician or FDA when making an approval decision. Just interested in your comments.
Yeah, certainly independent evidence is a very powerful thing and a very useful thing, a very convincing thing, but it comes at a cost, right? You have to study more patients, maybe for a longer period of time. We know that the success rate of phase 3 trials is somewhere on the order of 30%. So 40% of these studies don't work. And that means a whole bunch of patients are getting exposed to treatments that don't work, right? By necessitating this kind of independent assessment. Cumulative evidence, yeah, I understand the dependencies that build over time, but that comes at a benefit of perhaps fewer patients, quicker decisions to kill or to accept, or to kill a drug or to approve a drug, And so I think we need to be very careful about these kinds of trade-offs. And if we set the bar really, really high of this independent evidence, p-values less than 0.05, two adequate, well-controlled trials, who knows how many type 2 errors there are, and we do know how many phase 3 trials fail with the exposure of patients, et cetera, in an attempt to get this independent evidence. So there's costs and benefits to both ways of thinking, but it's a good question and one that we as a society, a regulatory community, and drug development community need to grapple with.
There's another comment about regarding equipoise. Seems to me that the neglect of the applicable utility functions leads to apparent disagreements between equipoise and prior probabilities. If many people would benefit from marketing authorization, then proceeding to phase 3 could be justified even if the prior probability of a meaningful benefit could be small. So decision analytically, equipoise is not needed.
I love that observation and that comment. I had not thought of it in that way. Melissa, I hope you can cut and paste these comments and questions into
I
a
will
document
save these comments,
share
yes.
it, because that is an excellent comment, and one that pharma companies do tacitly, right? They're looking at the market size, the utility, et cetera, but one which regulators only do passively, right? There's times when drugs have been approved because of the unmet medical need, and I won't point out bad examples of this, but one good example is sacubitril valsartan, or Entresto, for patients with preserved ejection fraction, right? They didn't get a p-value less than 0.05, but the unmet medical need was huge. The cumulative totality of evidence of it working in patients with reduced ejection fraction, et cetera, led to an FDA approval. So these things are done in some tacit ways or heuristic or intuitive ways, but I think we could bring some value if we brought them to the fore and built those utilities into our whole framework. All right, Melissa, I'm gonna maybe I'll be able to answer some of those others via email or in other ways, but I'm gonna get through the last few slides here, okay, with the epistemology. Okay. So for those of you who don't know, epistemology is the theory of knowledge, the study of how we accumulate knowledge, especially as it says here, regards to methods, validity, and scope. So it helps us distinguish justified belief from opinion. So it helps us, how do we know what we know, right? Epistemology is about the methods, and in a sense, statistics to me is really very strongly connected to epistemology, right? How do we believe? What is likely to be true? How do we infer this? What methods, validity, scope, et cetera, helps us distinguish our beliefs from our opinions, right? I like to say statistics is the science of discerning what is likely to be true. And in that sense, you can see it's aligned with epistemology. Now, one of the things from ICHE 9 that we all love and buy into is the primary analysis has to be planned a priori. And we always say that the degree of confidence in the final results and the conclusions of the trial depend on the extent to which we actually pre-specify what we're going to do and how we're going to do it, right? We just can't make up analyses as we go. We all understand exploratory analyses should be taken with less confidence, with less belief, right? And we've stated it in this way, this concise sentence in ICHE 9, right? Well, the question I have is, where do we focus our a priori planning? On just the new experiment or trial that we're doing? Or should we focus on the whole paradigm of what we're trying to show or believe, right? In the frequentist assessment, we compute p-values, we pre-specify what we're going to do for a single trial, and then we do a post hoc assessment of how it fits into other evidence. What critical value could we have used in the spirit of layman and how we report p-values these days, right? And we ask ourselves, is it consistent with previous findings? We all know these things are subject to bias. Once you see the data, you can make up all sorts of stories and justify the results, good, bad, or indifferent, in many, many different ways, right? In the Bayesian approach, we quantify the belief a priori, and in a sense, our pre-specification is related to the whole system. What is your prior? What is your analysis plan for the current study? And you're taking a bigger view of pre-specification so that the credibility of the posterior results once you work through that machinery, you can stick with that or have greater credibility about what the results are. There's less post hoc assessment about the probability that the drug works because you've pre-specified the entire framework for inference. And this is one of the reasons that I think Bayesian approaches are underestimated and could be much more powerful, is that epistemologically, they take the whole framework of evidence and decision-making into account and don't do the post hoc. Well, you're always going to do some post hoc assessments, but the frequentists are subject to much more post hoc assessments about how to incorporate present findings with others, okay? P-value is an ultimate test statistic. It's nothing more than a test statistic. It's a precise answer to the wrong question. It's even a poor answer to one of the three important questions of inference, right? A Bayesian probability might be considered a vague answer because you have a subjective prior, but at least it's to the right question. It's what scientists want. It's what all of us want. I think using the upper bound on the posterior probability of the null hypothesis being false is a very simple and powerful way to go. At a minimum, using this point prior for the hypothesis and the Bayes factor bound, I think this could be extremely useful, and I encourage you to use it when you read papers and don't have access to data. Go through that mental exercise yourself. Significance level and power are needed to design a study. That's the frequentist approach, but positive and negative predictive values, that is Bayesian posterior probabilities, are needed for interpretation. Everybody knows this for diagnostic tests. Everybody believes this, thinks this way, works this way. So why don't we do it for clinical trials, right? We should. Bayesian perspective answers the question of interest. I believe statisticians should convey on a regular basis the likelihood that a hypothesis is true or false. Stop doing the bait and switch. Stop giving a p-value as an approximate answer to this Bayesian posterior probability. It's why I was one of the 800 signatories of this paper in Nature, Retire Statistical Significance. I'm not saying, and they did not say, get rid of p-values in hypothesis testing. It's simply a matter of it's only part of the story. And in fact, I suspect many of you have heard in your careers this quote from the esteemed John Tukey: far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can always be made more precise. When I tracked this down, I had trouble tracking it down, but when I actually tracked it down, where it came from, the sentences leading up to this, I thought were even more important, because he said the most important maxim for data analysis, and one which many statisticians seem to have shunned, is this. And he said this in 1962, published in the Annals of Mathematical Statistics, where he was basically chastising all the mathematical statisticians for doing fancy mathematics, but forgetting that the real thing we're trying to do is get an answer to the right question, even if it's approximate. And he said, it's the most important maxim for data analysis. And he says many statisticians seem to have shunned it. I think there's some truth to that to this day, that people are still trying to get the right model and assumptions and trying to get a very precise p-value, shucks, to the wrong question. So what I like to say, because Bayesian approaches are epistemologically superior for all the examples and reasons I've given, I think that survival of the fittest dictates that the Bayesian approaches will win out over time. And if you look at the evolution of man over time, you will see that he is becoming more and more Bayesian in his thinking. And with that, I'll say there's some additional readings. This article, Détente, comparing and marrying p-values and Bayesian posterior probabilities, is out there. And the probability I thank you is 0.9999, which actually is the same as 1. The probability you thank me, well, I'll let you develop your own posterior distribution of thanks on the Mother Teresa gratitude scale. But thank you for sharing the last hour and a half with me. I appreciate your time. I hope it gave you some new insights and thoughts about the importance of Bayesian thinking and how I think about it. All right, Melissa, back to you to wrap up, or to, if anybody can stay on, or if you, whatever, to chat a little bit more if need be.
Yes, thank you very much, Steve. I think I will say thank you. My probability is also that 0.999, essentially 1. There are a few more comments that are coming in through the chat, primarily to say great presentation and saying thank you. I will save the chat and then make that available in case there's anything else you wanted to respond to directly. But we are at time, and so I will be considerate of people's time. But thank you very much again, and thank you all for joining today.
All right, Melissa, thank you, and to all the Bayesian Statistics Working Group and the entire audience, my deepest appreciation. Thank you very much.
Thank you. Have a great weekend.
Okay, bye.